Good afternoon and welcome. I want to thank you for joining us today for the third Agro Lunch and Learn webinar of 2022. This webinar series is made possible through the very generous support by Eurofins Agriscience Services, and we want to thank them for their continuing support. I'm Tom Sparks from Agrilucent, and I'm a member of the webinar committee and the executive committee for the Agro division of ACS, and I'm the moderator for today. If you're attending this for the first time, you'll be able to submit questions and comments for the speaker via the Q&A tool on your screen, and all the questions we'll address at the end of the presentation. If you're not a member of the Agro division, we encourage you to join it. It's a modest cost every year, but it supports a lot of programs, and more importantly, it supports programs like this. And just as another reminder, if you're not already aware of this, is that the fall ACS meeting is coming up in Chicago from the 21st to the 25th of August. And registration will be open in mid-June. Hotel booking is already open. And for this meeting, there'll be two types of presentations at the Chicago meeting, live and on-site and live remote. There's not going to be any recordings, and Agro is going to support both of those. If you have any questions, contact Heidi Ihrig. If you have any questions about the meeting or Agro's participation, et cetera, or what symposia are planned. Today, I have the honor of hosting Dr. Frank Wessels. Frank is currently has been since he graduated with his PhD in 2010 from the University of Florida in entomology. And right after graduating, he joined Dow AgroSciences, which is now Corteva. And when Frank joined, he joined in the insect management group of Discovery Research, and it's where he's been since 2010. From 2017 to 2022, he was actually group leader for the insect management group. At present, Frank has assumed a new role, which is, and I have to read this correctly, Global Insecticide Resistance Management Expert for Crop Protection Discovery and Development. And he's part of the Corteva Discovery Mode of Action Resistance Center of Expertise. Frank has numerous presentations and reports. He's got at least 9 patents that we can count so far. And importantly, for the purposes of this presentation, Frank has been a member of the Insecticide Resistance Action Committee for a decade now, and is currently chair of the IRAC, Insecticide Resistance Action Committee, IRAC Mode of Action Working Group. And with that, I will turn it over to Frank.
All right, thanks, Tom. Let me share my screen here. All right, how's that look?
Looks good.
Okay, so Tom, thank you for the very kind introduction. I appreciate that very much. And I'd also like to thank the ACS Agro Lunch and Learn Committee for the invitation to come and speak today. I really appreciate the opportunity to kind of maybe assemble some of my thoughts about recent trends in the insecticide discovery area and innovation kind of that's gone along with that. So let's see. Let me select my screen here. All right. So when I was doing some research on this and kind of looking through the literature on innovation and what other scientists have said about innovation, there are several reasons that have been listed for why we would want to have innovation in insecticides. And so among those, sustainable food production, improved productivity, so making the most of the land that we have available for agricultural production, replacing older technologies, and kind of along with that, I guess, lower application rates, increased selectivity, so better properties of insecticides, better integration or utility for integrated pest management, and then of course for insecticide resistance purposes and resistance management. So this kind of dovetails hopefully really nicely with last month's presentation by Dr. David Mota Sanchez on resistance, because I'll be talking about kind of innovation in the insecticide space and through a resistance management lens. So yeah, when Tom first approached me about this webinar, I thought of Steve Duke's 2012 paper on herbicide modes of action and why there's been kind of a lack of recent modes of action in the herbicide space. That's not to say that there hasn't been innovation in the herbicide space, but as far as new distinct modes of action, there are several years where there hasn't been any. And I encourage you to check out that paper. It's really well done and interesting hypotheses there of why that may happen. But I also came across just a recent publication on the same topic by Stephen and a colleague. And even here, 10 years later, the story is kind of still the same as far as not a lot of new modes of action and herbicides being discovered. So that's not to say to really speak bad about kind of herbicide discovery, but more to say that really we should not take advantage and assume that things will always be seen in innovation, because really the story I hope to share today is that there has been quite a bit of innovation in the insecticide space, but also something we need to manage appropriately to make sure we're not losing that to resistance and for other reasons. Okay, so to start out and talk about some definitions, I wanted to touch on IPM and IRM. So IPM, if you're not familiar, is Integrated Pest Management, and it's the integrative practice for sustainable pest control. Similarly, so actually I came across this diagram, which I really liked from Dara in 2019. Great paper on this. I like this because it's a lot of definitions and descriptions of IPM focus more on the pest management side, like the tools and things available. This is more of a holistic view where you see kind of some of the key driving concepts behind IPM. It includes some of the key stakeholders with consumers, producers, and then also things that are part of the IPM solution, so communication, planning, and organization. So some of these tools for the pest management, there's chemical, microbial, physical, mechanical disruptors. I'll talk about some of these today, yeah, and having access to all of these different technologies is important for insecticide resistance management because it's an essential component of IPM. So IRM is just a strategy to delay or prevent the development of insecticide resistance. But in order to do that and to have effective IPM, it really relies on access to a variety of effective pest management tools. And that's kind of here in the knowledge and resources, making sure that for whatever particular crop or pest in that crop, that there are solutions available. And the reason that's important is if we have a hypothetical resistance management program here, so this is just a corn crop with a hypothetical pest that has multiple generations during the growing cycle of that crop, and they're divided into windows. And so each one of these windows represents a discrete pest life cycle for this hypothetical pest. And each window would be a time where if you're a grower and the pest is present in sufficient numbers, you would want to potentially use a control technology. So in this example, if you had access to two control technologies, but we would definitely not recommend using them sequentially. So using the first one, repeating that, and so on. Because a key element of effective resistance management is to alternate technologies, especially mode of action classes, to prevent or delay the development of resistance. And even better than having two options, as I mentioned, would be to have as most options as possible. So if you could rotate 3 options, that would even be much more preferred of a strategy from a resistance management perspective. Okay, so Tom mentioned my involvement with IRAC for many years, so I thought I'd give a brief high-level introduction of the Insecticide Resistance Action Committee. because a lot of the talk today is put together through the lens of IRAC and the mode of action classification. So IRAC was formed in the mid-'80s as a specialist technical group of the Industry Association CropLife. And our mission is to facilitate communication and education on insecticide resistance and to promote and facilitate the development and implementation of IRM strategies to support agriculture and improve public health. So this is CropLife International, and there's multiple racks within CropLife International: fungicides, insecticides, herbicides, and rodenticides. But within IRAC, we have in the international team, we have the executive committee and steering team, and then multiple working groups. So the mode of action working group that Tom mentioned I'm involved in, outreach working group, a test methods group that develops test methods and approves them for baseline monitoring and resistance monitoring. And then biotechnology, public health, and then the crop protection team is divided based on target pests. So you can see those there. And we often say that resistance, you have to think of it globally, but also you're ultimately acting on it locally. And so there's multiple IRAC country groups that are focusing on translating a lot of those IRAC materials to local languages, providing resources and tailoring resources to be more specific to problems in those specific countries or geographies. All right, so speaking of resources, we have several resources available. Teaching materials, test methods, posters, white papers. And as was mentioned last week with Dr. David Mota Sanchez, the Michigan State University Arthropod Pesticide Resistance Database, IRAC provides financial support because really it's a huge undertaking to maintain that massive database, but it's such an important resource from a resistance management perspective to have. And so we're happy to support that because there's really nothing else like that available anywhere in the world. And then finally, what we're probably best known for is the mode of action classification scheme. To talk a little bit about that classification scheme, it's really the preeminent source for global insecticide mode of action classification. So it's utilized by a lot of regulatory agencies globally, and it's a living document. So it's constantly changing because it leverages the most up-to-date research and information available for classification. But we intend for it to be a simple, straightforward mechanism for classifying insecticides, acaricides, and nematicides for IRM programs. So really the kind of high-level view of the mode of action classification, or IRAC in general, kind of our goal is to take this big, kind of complicated technical picture that is admittedly pretty difficult to follow if you aren't pretty familiar with it and the literature, and taking this classification that's representing literally thousands of modes of action, or thousands of, active ingredients, and how do we get that information to the end user, to a grower or farmer that's going to use that information to make decisions in their field. And so the way we do that is through our label recommendations. And so we recommend putting, this is just a generic hypothetical product label for an insecticide, and we have this classification label recommendation where it says group, and it has a number representing a mode of action group and a letter representing a subgroup if there are subgroups for that group, and then whether or not it's an insecticide, a fungicide, or herbicide. So an end user doesn't need to be familiar with the thousands of different names and whether that's a pyrethroid or a carbamate. All they need to know is, for their crop and pest, if they, in their plan, if they used a group 1A insecticide for this example, in their next application, they would want to use pretty much anything that's appropriate that's not a 1A insecticide, preferably another mode of action group, so say a group 3A insecticide, something like that. Okay, so this is a high-level view of the IRAC mode of action classification scheme. I know it's a little bit hard to see, but kind of the structure of the classification is you see these different boxes. This is the poster. We also have a white paper and even an app now But each box represents a distinct mode of action group. If we blow up one of the groups here, the group 2, you can see there's different subgroups within this that represent different structural classes of insecticides within this group. Also, you'll notice the poster's pretty colorful. This is to identify what the targeted physiology for the different mode of action groups is. So whether it's targeting nerve and muscle, tissue, growth and development, respiration, midgut, or whether it's unknown or non-specific mode of action. Okay, so when we think about changes, I wanted to rewind about a decade and see what things looked like 10 years ago. And this is the poster that was launched at the end of 2011, so effectively the 2012 poster. And what we note is really it's very similar. If you're not too familiar, it's hard to pick out specific differences. There's still a bunch of boxes, a lot of hexagons, some divisions within there. But while it's not immediately apparent that anything has changed, quite a bit has indeed changed. So we look at the number of mode of action groups over the several years of the existence of IRAC. You can see around the year 2001, there's a steep jump in number of mode of action groups. So that isn't because of any innovation at that time point. That was because the initial classification was really only encompassing acaricides. And in 2001, insecticides were added to the classification. So this is kind of really the start of the mode of action classification scheme as we know it today. You can see there was some increase in the number of mode of action classes over several years, but if we go back and look at the past decade, and really since 2016, there's been almost an inflection point where we've seen several new mode of action classes introduced in recent years, which is an exciting turn of events. Similarly, if we look at the subgrouping classifications, same thing starting kind of in the 2001 with establishment, they've gone up and down as that the classification has evolved. So some groups were divided and then coalesced, or new ones created as new information has become available. But really over the past decade, you see a consistent increase in the number of new mode of action subgroups. Okay, so I mentioned wasn't really apparent, but a lot has changed in the past decades. And some of the things that have changed were biologics have been incorporated into the classification scheme. 7 new mode of action groups, which is 21% of the total. 13 subgroups. 33 new active ingredients were classified, and the incorporation of nematicides. And so I'm not going to speak to nematicides for the rest of the talk. It's a separate classification scheme. There's a classification scheme and a resistance risk statement. So if that's something you're interested in, you can look at the IRAC website under the nematode team and download any of these documents. But focusing on the top 4, you see these kind of icons. So I'll be using these icons to kind of bucket the slides, but I'm going to go through kind of each one of these points and talk about the biologics, new modes of action, and so on. Okay, so for biologics, there's a variety of definitions. I was doing some research to kind of identify a common definition. I couldn't. Biopesticides, biologics, biologicals, a lot of authors and researchers kind of lump these things together differently. And really, I'm not an expert on this, but some of the things that are classified as biologics are really out of scope for IRAC. So things like repellents and pheromones, and even biological control agents, predators, parasitoids, things like that. Really, that's not something that really fits with the philosophy of the classification scheme from a resistance management perspective. They're absolutely great tools and important from an IPM perspective, but not really congruent with the classification scheme. So for the purposes here, I'm using biologics as more of a definition to broadly define and encompass things that are new or different or innovative compared to traditional chemistry inorganic compounds and natural products. A lot of people lump natural products in with biologics. I'm not, simply because biologics or natural products have been a part of the classification scheme really from the beginning, with like nicotine and permethrin, things like that. So I fully expect the classifications and definitions to evolve over time, but some of the things that I'm considering to be a biologic are like viruses, peptides, nucleic acids, microbes, botanical extracts, and even mechanical or physical disruptors, so like diatomaceous earth or mineral oils. Okay, so moving on to new mode of action groups in the past decade. Like I said, since 2016, there have been 7 new groups. If you look at the poster, the new poster, it's kind of encompassing all these groups and a little bit more, and I'll talk about that in a little bit. But interestingly, almost half of those most recent groups contain exclusively biologics, which is a trend that's new and interesting. And these aren't to say that these were just added because we started incorporating biologics. These are actually kind of newer technologies to the marketplace. So I'm going to go through and kind of touch on a lot of these individually and give kind of a Cliff's Notes version of each mode of action group that's new. Usually from an IRAC perspective, we try to avoid mentioning specific active ingredients or products, things like that, because we don't want it to be construed that we are making any recommendations or kind of preferentially kind of suggesting that maybe one thing is better than the other. So just to say that, I'm not saying that, I'm just looking at all these active ingredients from kind of a holistic perspective regarding innovation. Okay, so the first new group in 2016 in the past decade was the Corteva organ modulators undefined target site. Really, this is a whole story, and to get into the story, you have to understand the key players and where they were originally. So in 2012, there was Group 9, which is listed as the selective homopterin feeding blockers with pymetrozine and flunecimide. And then pyrofluconazone was categorized as an unknown mode of action at the time. So fast forward to 2016, when flunecimide was broken off and added to Group 29 and designated the cortitonal organ modulators with an undefined target site. And the way this came about, I think, this is a really good example of the mode of action classification as a living document where changes are made with new information becoming available. But it was interesting because pymetrozine, even in 2016, was a pretty old molecule. It was first launched in the mid-90s. Flunecimide was launched in the early 2000s, and pyrofluconazone in 2010. So it was interesting to see kind of this reshuffling. It kind of goes to show sometimes how long it can take to determine specifically what the mechanisms of action of these certain active ingredients actually are. So pymetrozine was first linked to cortitonal receptors, which were part of the names in 2005. So there's a study in Journal of Experimental Biology looking at electrophysiology and symptomology of pymetrozine on the migratory locust and kind of linking pymetrozine to the action on cortitonal receptors. But the mechanistic target site wasn't really identified until 2015. And in 2015, this paper was published in Neuron, where the Nesterov and colleagues looked at pymetrozine, pyrofluconazone, and deacetylated pyrofluconazone. And I need to explain, so pyrofluconazone is a prodrug. When it's dissolved in aqueous solution, it's just spontaneously deacetylated. So they were including this kind of as a control to evaluate that as well as the parent compound. And they identified TRP channels, specifically a TRPV channel, as the target site for pymetrozine and pyrofluconazone. Kind of what that receptor was identified as is a heteromer of 2 proteins named Nanchung and inactive. So they express these proteins in CHO cells, and this is showing fluorescence, so calcium fluorescence in the CHO cells when they're exposed to these different active ingredients. You can see with just the parental cells, you only see background fluorescence. Similarly, if you express Nan-Chong or inactive alone, you also only see the background fluorescence. But once you combine those two peptides, you see really good activation with all three of the active ingredients that were tested. So showing that that it requires both of those proteins to form a functional channel. And because of this, pymetrozine and pyrofluconazone were subsequently assigned to group 9B. If you're wondering why there's no 9A, that's because 9A used to be cryolite. We typically don't reuse mode of action groups to prevent confusion with insecticide labels and things like that. So that's why there's only a 9B and a 9D, because 9C was flonicamide, which then was moved to group 29, which was created. So group 9 was renamed the corticotonal organ TRPV channel modulators, and group 29, of course, is the undefined target site. And then avidopyropin here was assigned group 9D in 2017. Okay, moving on to the next group, group 30, the GABA-gated chloride channel allosteric modulators. So these are the metadiamide and isoxazoline chemistries, and their discovery stemmed from research on the anthranilic, the phthalic and anthranilic diamides. So this is flubendiamide, a phthalic diamide that's acting on ryanodine receptors. And you can see that the two amide groups off this central ring substituted in the ortho position. And in broflanilide here, you can see that amide, they're kind of similar molecules, but the amides move from the ortho position to the meta position, so it's hence the name. But interestingly, while that doesn't seem like a very big structural change, that led to considerably different symptomology on target species. So instead of seeing complete contraction and paralysis, which is pretty characteristic of ryanodine receptor modulators that are affecting the muscles, they saw excitation, convulsions, and paralysis, which it was noted was similar to GABA antagonists, such as fipronil. So in this study from 2013, Nakao and colleagues characterized metadiamides on mutant Drosophila GABA receptors expressed in cell lines. And what they saw is with the wild type represented by these circles, both broflanilide and fipronil had a dose-dependent inhibition of the receptor. But this mutation, 2' in the 2' and 6' positions, so these are amino acid positions in the channel of the receptor. And this, so if you look at the kind of square bars, broflanilide still showed activity on these mutants, whereas flonicamide lost efficacy on those mutants. And kind of in the contrast, they noted that this other mutation, G336M, caused a loss of function for loss of activity to broflanilide, where fipronil still retained activity. So it suggested that they were acting at different binding sites and supported the creation of a new mode of action group for allosteric modulators. There's also some really nice computational chemistry done in the Nakao paper, so I encourage you, if you're interested in that story, to go check that out. All right, our next new group is the first biologic on the list. These are baculoviruses. And so the way these work is that there's these occlusion-derived virions that are packaged in different occlusion bodies. And so those occlusion bodies can have different morphology. So that's how you get the designation of either a nucleopolyhedrovirus or a granulovirus. But they're applied just like any other foliar crop protection product. It could be sprayed on a plant. Your pest ingests these occlusion bodies, and then they are dissolved in the midgut, and those virions move past the peritrophic membrane and enter the midgut, where they begin to replicate. Then they cause a secondary infection where they enter the hemolymph and go throughout the insect and infect other cell types and other tissues, leading to ultimately the death of the insect and the spread of more occlusion bodies. The insects usually kind of die in a way where they serve to spread more virus in the area, which is kind of interesting. So it was a unique and well-defined life cycle for these viruses and supported the creation of a new mode of action group. And you can see they're classified based on the target pest that they're and whether they're a granular virus or a nucleopolyhedra virus. All right, next, another biologic, the group 32, nicotinic acetylcholine receptor allosteric modulators at site 2. So one thing you'll notice is we don't like short names, but kind of complicated. But this is a peptide toxin from spider venom. It actually came from this funnel web spider from Australia. Really, whether I guess depending on your perspective, really cool insect or cool spider or really creepy one. But the specific protein is the GS omega kappa hexatoxin HV1A. And so being a peptide, penetration into the pest target insect is a challenge. And reading up on this is interesting that in Drosophila suzuki, so spotted-winged Drosophila, has shown that you get better mortality when you co-apply it with adjuvant, so an organosilicon adjuvant, silhouette. Similarly, some of the products targeting lepidopteran pests require tank mixing a Bacillus thuringiensis product to facilitate gut penetration. These are pore formers in the gut. So kind of two creative solutions to get the peptide into the target insect. But the reason I mentioned them is I just thought it was kind of interesting that we're having to come up with creative solutions because the spiders obviously evolved their own unique solution to get the toxic peptides into their prey. So intoxication symptoms of the hexatoxins are neuroexcitatory, and they were characterized as positive allosteric modulators of the nicotinic receptor because of their high affinity binding, which was enhanced by nicotine and other agonists. But the spinosins are also allosteric modulators of nicotinic receptors. They are in group 5 on the classification scheme. And so researchers, they published this paper in 2019 where they wanted to see are these acting at the same site. So they did a competitive binding experiment where they tritiated dihydrospinosin A and tried to displace it with either spinoside or a representative hexatoxin here. And you could see dose-dependent displacement with spinoside, but the hexatoxin didn't displace spinoside. In the kind of contrasting experiment where with labeled hexatoxin, you could see dose-dependent displacement with the hexatoxin, but no displacement with spinoside. So that suggested that these were binding at unique allosteric binding sites. And group 5 was renamed as site 1, and group 32 was created for the hexatoxins and designated as site 2. Okay, so for group 33 and 34, these are calcium activated potassium channel modulators for 33. And the active ingredient in this group is acynonapyr. And for group 34, these are MET3 inhibitors, but acting at the QI site, represented by flumetiquin. So both of these are very recent products, just launched in the past few years. And so because of that, there isn't a lot of information that's publicly available. So the mode of action characterization information, at least to my knowledge, isn't published. I guess this highlights the cutting edge nature of the IRAC classification scheme, but also when an individual or usually company comes to the IRAC classification or IRAC mode of action group and requests a classification, any supporting data that they share is kept confidentially within the group. So all of the information I'm sharing today is just publicly available in the literature and not sharing anything. So hopefully we'll see something on these two compounds and the mode of action in the near future in the literature. Okay, and then our last new group, mode of action group, is group 35, the RNA interference mediated target suppressors. So the 35 is in parentheses here because This only appears in Appendix 6 of the IRAC Mode of Action Classification Scheme. If you go on the IRAC site, you can download the white paper version. That's usually the most up-to-date version of the classification scheme. And if you scroll all the way down to the bottom of that, the end of that is Appendix 6. So you'll see compounds that have been classified by the mode of action team, but they won't show up in the classification until they are registered for use somewhere in the world. So RNAi is a natural biological defense mechanism in most eukaryotic organisms. There's been a lot of information and a lot of kind of reviews on how this works. So I'm not going to go into it, just to say that double-stranded RNA leads to degradation, specific degradation of mRNAs and ultimately protein suppression. And the active ingredient that was classified by IRAC was a sprayable dsRNA named ledprona, and it specifically targets Colorado potato beetle, and specifically this proteasome subunit beta type 5 that's a protein involved in a pathway for removing damaged protein from cells. So there's a couple of good publications on this that show where they link kind of a dose-dependent effect of the double-stranded RNA on the beetle mortality, and then also where they show that exposure to that double-stranded RNA does cause a reduction in the targeted mRNA and ultimately protein. So linking the exposure to the double-stranded RNA to the mortality, and therefore kind of supporting that this should be a novel classification group. Okay, so moving on to new mode of action subgroups over the past decade. I mentioned there's been a pretty consistent increase over recent years, so I kind of highlighted these out on a little bit of a map. So Bacillus sphaericus was added to group 11 as 11B in 2012. 2014, the butanolides were added to group 4 and 4D. And then along with group 4 in 2016, there were the mesoionics added as a unique subgroup. And then just recently, the pyridines, I always have trouble saying that, were added to subgroup 4F for the nicotinic modulators. And 2015, there's bifenazate was added as a new subgroup to 20D, and then the methyl isothiocyanate generators were added to 8F. And if you look at this increase, in 2019, there was a sharp increase in the number of subgroups, and the reason is because of the incorporation of biologics. So I highlighted some of the specific biologics that have defined mechanisms of action or modes of action, but several are unknown. So these are classified in the unknown classification group, but receive subgroups for bacterial agents that are not BT, botanical essence or extracts, fungal agents, and then mechanical or physical disruptors. And there's also two more subgroups for peptides and viral agents that are not baculoviruses. And these are in italics because there's currently no active ingredients that are represented by these two groups, but the mode of action classification teams established these just for future technologies that could be developed. So over the past decade, the growth of the subgroups has been most prominent in group 4 and with the unknown group because of the incorporation of biologics. And finally, looking at new active ingredients that were classified in the past 10 years, a diversity of new additions over the past decade. These kind of colored boxes behind the hexagons indicate active ingredients that are grouped together in the same mode of action group. And then in the boxes here are two technologies that are in the appendix, so they have not been registered yet for use anywhere. So they're in the appendix of the mode of action classification. We look at the origin of newly classified active ingredients. You see about half are still traditional synthetic organic chemistry for their origins, and about half are the biologics. So that's heavily influenced by the addition of several technologies in the unknown group, but there are some new ones as well that I highlighted with the hexatoxins and the viruses and so on. And so also, at least to my knowledge, only one semi-synthetic natural product, aphidopyropin, in the past decade was introduced. And then also, if you look at the targeted physiology, a broad diversity there as well for these active ingredients. So about a third targeting nerve and muscle action, and about a third that have unknown modes of action or non-specific, so like multi-site type chemistries, but heavily influenced for the unknown, again, with the biologics. But then there's representatives that are affecting respiration, the midgut, growth and development, and a new classification or new physiology classification that'll be added once a RNAi product is registered, and that's the targeted protein suppressors. Okay, so a few years ago, Tom and myself and some colleagues published a paper on what we were calling the new age of insecticide discovery, and I think it's still it's relevant to the discussion today. So early on, I'd say in the early like 50s, 60s, and so on, really the majority of sales for insecticides in the United States or globally was only due to three primary modes of action, represented by DDTs, cyclodines, and the carbamates. You can see the use of those or their kind of control of the market has decreased over years. And since then, we've had a huge number of new modes of action that were added to kind of the global toolbox of insecticide technologies. But as these, and it's interesting, the trends, because you see some kind of big groups with the neonicotinoids, the pyrethroids and diamides, but this other group is one that's kind of taken over and dominated. When you kind of dig into that a little bit more, in the early days of insecticide discovery, which has been coined by John Casta and some others as the golden age of insecticide discovery, you had these major classes of insecticides, so organophosphates. And I mean, keep in mind, this is a log scale, so the number of organophosphates, it's pretty mind-boggling, but there was a lot of them. The carbamates, several of those within that class, synthetic pyrethroids, a lot of synthetic pyrethroids were developed. And so what we've seen, this is kind of pre like 1990, but what we've seen since then is kind of a different strategy, if you will, where there's been a increased breadth of classes of insecticides. And so when we say classes, these aren't necessarily distinct mode of action groups, but we bucketed these based on kind of the discovery classes, looking at synthetic chemistry, natural products, things like that. So there's been an increased breadth, but the number of active ingredients within each one of those classes hasn't really taken off to the level that it had in the early days of insecticide discovery. So that's kind of an interesting shift in trends. And hopefully I've convinced you today that the recent trends suggest that maybe this new age is currently evolving to include a variety of new technologies like the biologics. And so only time will tell if that trend continues or even expands over the next several years. So to summarize, the discovery of, I'm taking these there's a paper on IRAC published by Ralph Nowen and colleagues in this book, Modern Crop Protection Compounds, a few years ago. So I'm kind of stealing a lot of these summarized points from there, but really I think they hit the nail on the head. So I didn't really talk about costs of development, but discovery of novel insecticides is costly and very challenging. So it's, while we've seen a lot of innovation in insecticide discovery, we have to realize that that really came out of a lot of work over many decades with many hundreds or thousands of different scientists involved in bringing technology to a new technology to the market. So because of that, it's essential that we work to maintain the efficacy of insect control technologies, because really pest susceptibility is a highly valued commodity, and it should be seen as everybody's shared responsibility. So because it's far easier to manage and delay resistance than to regain susceptibility. And also just put a plug out there that we need to be open to new technologies and innovations, kind of just broadly as consumers, regulators, industry representatives, all of that. And as they say in the stock market, past performance doesn't indicate future success. So that's very true here. We shouldn't rest on our laurels and assume that there's always going to be this innovation. So we need to act as if show that these are kind of the very important tools that they are, and use them appropriately. So with that, that kind of wraps up my talk, and really I'd be happy to take any questions if there are any.
Thank you very much, Frank, for a really cool talk. Raised some really interesting questions and provide a lot of really good background information. We've got a couple questions. I'll take the second one. Could you share this PowerPoint for everybody that's tuned in? This is being recorded, and so a version of this will be gone through and kind of edited a little bit and then made available to anybody that is tuned in today. So that's going to be out there in a couple weeks. There's a question that, and if you've got any other questions, the Q&A button at the bottom, you can type in your question as we go, because we still have a few minutes. Other question is, as we evolve from biologicals characterized at the species level to biologicals characterized by genomics, how do you see the evolution of groupings?
Yeah, that's a really good question. So, over the years, there's been a few attempts at classifying insecticides for different purposes. But I think from a resistance management IPM perspective, focusing on mode of action, that's going to be critical to kind of focus on that. I'm not sure exactly what's meant by characterized by genomics. If you're talking about targeting specific proteins or peptides, honestly, I think we're going to have to wait and see, because really we're going to have to talk about those and the implications of classifying. All the classification is going to be kind of done in the light of for resistance management in that context. That's kind of the overarching theme. So if it makes sense based on that, and then also keep in mind things. It's a living document. So if new information comes available, or we see kind of issues, the unexpected issues that popped up, that could change the classification scheme as well. So hope that answered the question.
Yeah, I have some other ones. In 1990, 30 years ago, John Cassida published an article in a book that he edited called Pesticides and Alternatives. And the premise of this article was that there was a finite number of modes of actions for insecticides. Given that this was 30 years ago, I'm curious, in the intervening time, what your opinion of this comment is, one way or the other.
Yeah, that's tough. I guess I think the data would support that. There has been new modes of action discovered since then. So the data would support that there are potentially more. I don't recall the context of that. Like if he was just talking about like major mode of action groups or something like that. Yeah, I think the data, you got to be careful about making those statements, because you don't know what future technologies are going to bring to the table. But, and not just technologies within the actual active ingredient, it could be other technologies associated with it. But kind of from a high level perspective, there probably is a finite number of modes of action out there, but I don't think we've come close to finding all of those at this point. So I think there's still plenty of room for innovation. I think just the presence of so many companies and startups and agriculture and things involved with primary research on pest control, that kind of supports that as well.
Another question that's come up is, how about using nanotechnologies in the creation of new synthetic pesticides, nanopesticides, but also in the creation of, nanopesticides?
Yeah, absolutely. I mean, I don't think any technology should be off the table as far as development. That would be really cool. I think I don't really know, I guess, the full context about nanotechnologies, like what exactly that means, but I wouldn't rule out any new technology. And honestly, there's probably somebody out there working on this or thinking about it, either in an academic setting or in a startup somewhere, or even in a big company, who knows. But really, I think, with the importance of agriculture to our society, just our existence, and the kind of finite resources that are available, we should have all appropriate kind of sustainable options on the table for managing pests in agro ecosystems.
All right.
Good question.
Based on your presentation, we still have a few minutes. It appears that the recent focus has been on biologics. And it's important potentially from a new mode of action perspective. Do you see, from both what you see on IRAC and from the larger crop protection arena, do you think that this trend will continue?
Yeah, good question, Tom. So I think so currently. I didn't share a slide. I did put one together just looking at the costs. It's kind of hard to nail down the cost of development, but there's a lot of costs involved in discovering new modes of action, traditional chemistry. And I think currently the data supports that it's a lot cheaper to establish to develop a new biologic currently. And there might be some incentives from a regulatory perspective in different geographies globally. So because of that, there's probably an increased incentive economically to develop biopesticides and biologics. And so I think you're going to see that trend continue. But I don't know kind of at what pace or how quickly that would evolve. But I do expect to see more in coming years, for sure.
There's a question I'm going to ask the person that sent it if they could elaborate a little bit. What about for fipronil using? And I'm not sure what they're asking. To the person that sent that, if you could provide a bit more detail to the question, because it's not quite clear. Another one is, in terms of new insecticide modes of action, since you've been part of IRAC for a decade now, and what you've seen, what has been the most interesting or surprising, perhaps, from a mode of action point of view?
Yeah, from the mode of action point of view, I think the most interesting thing for me is getting a chance to see, for pardon the term, just how the sausage is made, kind of behind the scenes of. I really enjoyed the process. You probably saw that kind of how I put the talk together. Like I enjoyed the process and the discovery process and the determination process. So how do you figure out what these new technologies are doing, and how did people go about doing that in different ways? That's the part that I think is most interesting. I don't know if there's anything that was super surprising, but I do also like to see new technologies coming to light. That's really cool to see and to be a part of, I think, for me personally.
Cool. Another question is, and hopefully you can give some perspective on this, is what is the main difference between discovering novel insecticide modes of actions versus new herbicides with new modes of actions?
Yeah. So referring to that kind of Stephen Duke paper, there was a lot of other things involved other than just kind of the technical challenges. At least that's what he outlined in his paper, and I think he's correct. Talking about the influence of um, kind of herbicide tolerant traits and how that kind of decreased the incentives or interest um, in herbicide discovery for a period of time. And as anyone involved in R&D knows, it sometimes takes there's an activation energy it takes to kind of get back and gear back up into doing things at a very high level. But at a technical level, there are some, key differences. Herbicides, I know a lot of times there's several publications out there on this, the physical properties, insecticides too, but the physical properties have to be very specific, I know, for herbicides, because you're trying to trap, you want to get that herbicide into the plant and then get it to the growing parts of the plants, the meristematic tissue. So there are some limitations there that may not be necessarily limitations for insecticides, but yeah, other than that, actually, I think I believe there might even be a symposium at the ACS meeting this fall that's kind of exploring this, if I'm not mistaken. I've heard about that. So if that's a topic that interests you, and it is kind of interesting, keep an eye out for that in when the program's released, because that should be interesting as well.
Yeah, good perspective. Just as a side point on that, in Stephen Duke's article in 2012, he talked about the dearth of herbicide patents around herbicides because glyphosate had kind of solved so many problems for so long. Companies kind of didn't see it as a major market area. That's all changed now. There's lots of, if you look at the number of herbicide patents, they're on the increase. So maybe there's this lag phase that we're looking at this particular point in time.
Yeah.
Let's see. This is an interesting one. Do you know of any example where target-based discovery has been successful?
That is a good one. And honestly, I mean, Tom, you might be better positioned to answer this, but honestly, I don't know. I can't really recall any. It's definitely, if there are any, it's definitely not very common to my knowledge. I guess if you think about it from that's definitely a pharmacology or pharma industry approach to drug discovery, is focusing on the targets and working out from there. I think one of the advantages that we have in agriculture is that we can do testing on our target in the earliest stages of R&D. So and other people have mentioned that as well. It's not my idea, but almost if you think about it, like high throughput clinical studies, I think most people in pharma would really be very excited at that, to have access to that opportunity. So I think that's an advantage of agricultural discovery that isn't present in the farm industry. But in the future, well, actually, I just realized, but I guess the double-stranded RNA, the RNAi technologies, maybe I guess you could consider work from the target out as well. Other than that, I don't know of any. Do you know?
Yeah, that actually addresses the question that just came up: is RNAi targeted? And so, would you consider that?
Oh yeah. Man, we're all on the same page. Yeah. Thanks.
All right. With that, I think we're going to wrap things up. As a final point, I want to thank you, Frank, for your time and effort on this, and a really nice webinar. And I want to mention Eurofins Agri-Science Services for their continuing support of this. And I want to remind you that the next webinar is scheduled for September 14th. I don't know the exact topic yet, but keep open, keep watching your emails for information on that. And with that, I want to thank everybody that tuned in today for their attention. And again, Frank, thank you very much.
Yeah, thank you. Thank you, everybody, for hopefully found it interesting. So thanks.